So in this image, you can see the result of growth. You have to kind of visualize growth as it happens. One thing about plants as they grow is that they're growing onto existing structures. So I think we don't always see this illustrated very well. You see somebody kind of animate the growth of a plant. If you have an early growing part of a plant, you got, say, one leaf and a little bit of root. This plant goes on to produce additional growth. The same structures that were there in the beginning are still there, and they get built upon as this plant gets bigger and bigger. So I'm just going to keep kind of growing this up. We're moving in time from left to right. I got carried away with the roots there for a little while. Okay, so what do I want to emphasize here is that this location, this first leaf, is always going to be right there. It's not, with the exception of just a little bit of maybe some cell elongation, there's not really any new growth at this location. The new growth has to happen from the tips. And that's because as these parts of the plant get established, they get really fixed in place. Those cells that are juicy and squishy and elongating, after they differentiate, they become pretty fixed. One example that might resonate is we talked about xylem cells. They firm up with thick cell walls. They're dead on the inside. They're pretty much set in a shape that they're going to have for the rest of the life of that plant after a little bit of time. And there's just no stretching them out after a while. So the plant can't just, continue stretching from all parts. It has to leave the cells in place where they were made and build onto the parts of the plant that can still grow. So I guess maybe we stretch this out a little bit more. We can keep all those same leaves and so on. Keep growing. Reach for the stars as high as you can go. Grow some more root down. And so on. You guys get the picture. And then one possibility is that it's actually growing from one of those axillary locations that we saw in the last slide. So growing from here, from this tip, and then those axillary buds are kind of a location where there's some potential for new growth, that they might grow out to the side. Roots, in the same way as roots branch out, the tips of all these roots have growing potential. They can grow in directions, keep growing in that same direction, and they can initiate side roots as they grow as well. But what I really wanted to impress going from left to right was just the structure of this is all there, and new stuff gets added at the edges. All right, so here it is in place. Here we have our meristems again, and then this I like to think of as a nice kind of a way to think about plants growing themselves. So this is a time lapse of a skyscraper being built, and it's being built by having these cranes located on the top of the building. So a building obviously doesn't stretch, it's not biological, but the floors that were put in after they built 10 floors, they're building from the top. They add a new floor on top and so on. Very much like how plants are built. So once plants have structure put together, they're not going to change the shape of what they've already made. They have to add new stuff. All right. So this may be a little more detail than we need to cover, but just kind of on a cellular level, highlights one thing about the plant, apical meristem, where the cell divisions that generate these differentiating cells, if you think of every cell division as having one starting cell and two resulting cells, your starting cell has a stem cell character to it, And after one division, the stem cell has a daughter cell, and this cell basically remains a stem cell, and the daughter cell goes on to differentiate, as shown here. And that means that the sort of the stem cell of this, some of these cells stay as stem cells. So they add new cells below them, and that in turn pushes them up. So a lot of these cells are kind of always at the growing tip of the plant. As far as the structures they're going to put down as they're dividing and differentiating new cells, a lot of this we saw before when we talked about tissue systems. So we've got the, oh, sorry, I've got to fix. I think this is how it came from the textbook. Epidermis has kind of missed the mark on that one. So the outside layer of cells will be the epidermis. The cortex is this layer of ground tissue right here. We got some vascular bundles, and cortex is sort of an outer ground tissue outside of the vascular bundles. So like a ring of vascular bundles. And then the ground tissue inside of the vascular bundles is called pith. So a little bit of difference there. Ground cells here are pith, ground cells out here are cortex. Other ones we've seen, we haven't quite got to vascular cambium yet. That'll be a topic for the secondary growth. So stem, a little more commonly, a ring of vascular bundles. Roots, we saw some variation there, but in the dicot roots, this is more common to have the vascular cells, vascular system all in the center of the root and just having a cortex around the outside. So the ground tissue, mostly restricted to cortex. We'll see these kinds of cells and how they function when we talk about the root function a little bit later. That's a lot of what we covered previously. Starting organs from the apical meristem. So focus a lot on the shoot apical meristem. So the shoot apical meristem produces new stem and also new leaves. So how does this dome of cells, has this typically kind of a dome character to it? If you dissect away a whole bunch of the plant, you can get this little dome and see the leaves that are just being produced. So P1 and P2, these are the most recently produced leaves. P1 is just starting to look like a little outgrowth from this dome, and then P2 is older and it's a little more developed. It's kind of enveloping the shoot tip there and looking a little bit more like a leaf. Different kind of plant here with its little baby leaves. They call these leaf primordia, the starting leaves, just really tiny, but they're on their way to becoming leaves. Right next to the dome of the meristem cells. So as the plant is growing, its cells at the meristem tip are making a decision, kind of periodic decision for, all right, going to make stem for a while, stem, 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 stem. And then after some amount of time or some amount of growth, then a leaf is initiated. So this overall patterning is a collection of carefully orchestrated decisions in how much to grow this way versus when to initiate a leaf off to the side. So in this plant here, we've got some amount of time when it was producing only stem, and then it initiates a leaf off to the side while making more stem, another leaf off to a different side, and so on. So timing and positioning, these are carefully controlled in plants. And conveniently, I guess if you're trying to identify a plant, this is something that is pretty regular. So within the same species, you'll see the same kinds of patterns as far as where the leaves are made, how much distance is between the leaves, things like that are a little more commonly preserved. So the meristem, I mentioned that you kind of have to dissect your way down to get at it. On the left here, this is a meristem from a grass, and then a full-size grass over there. And the meristem proper is kind of the tip of the shoot, the stem portion here. So you often don't think about grasses having stems, I imagine. They do a lot of their growth next to the ground, especially our lawn grasses. But buried in there is a growing meristem tip. if a plant is going to continue making lots of leaves near the ground, then it's devoting a lot more of its time to making leaves and not as much time to growing the stem. And that's one strategy plants can have. They can kind of vary whether they're going for tall or leafy, leafy near the ground or tall with leaves along a stem. Two of the big strategies you might find in plants. And I mentioned here the concealed meristem. So here's a kind of an upshot of the arrangement of the leaves. As leaves are made, they're made pretty tiny to begin with, and they grow from there. And they can do this thing where they basically overlap in many layers, they overlap the meristem. And it's getting kind of a Brussels sprout look or cabbage look, if you're familiar with those vegetables. And they both have the kind of the same function, where the meristem here, this is very important cells. So they're the ones that are making new leaves and making new stems. And to a certain extent, they're irreplaceable, or they're not easily replaced. And so if they're concealed in here, they're also protected. So they're protected from, say, small animals who might have to chew through these new leaves to be able to get to the meristem. Protected against pathogens like bacteria and viruses. And it's kind of a better way to protect your meristem, while also this is really just a sort of a byproduct of how the leaves are made during development, is that they overlap the meristem itself. And this would be something sort of a general look to a bud. So a bud is made up of overlapping leaves with a meristem inside. So I mentioned the patterns are carefully controlled. This is an illustration of some of the different patterns you encounter in plants. And it's from a paper that was trying to computer model kind of what it takes to make these patterns. And I think what's neat about this is that the process doesn't have a brain to it. There's no like organizing signals that really permeate the whole plant. A little bit different than what we see in animals. What the plants are doing is really like a small set of cells are deciding among themselves, like, okay, at what point are we going to make a new leaf? Some of those cells kind of self-designate, like, okay, we're going to make a leaf. And their decision depends on the cells around them, and it depends on some organs that have been produced before them. So kind of a pattern of the next leaf is being made at a location that's some distance and some angle away from the previous leaf. So sometimes it's a little more one at a time. This leaf over here is the older leaf that was made some time ago, and this new leaf is being made at a position that's sort of as far away, if you look more at the center of this meristem, as far away as you can get from that first leaf. Other patterns can exist where you get two at a time. These are also kind of far away from each other. So this rotation of 90 degrees for this pair of leaves is a good use of the space relative to these other leaves. Also kind of fills in where the least, sort of the farthest distance from the previously made leaves. Sometimes you get three at a time. So relatively rare in the plants, but sometimes you get this where it's three leaves at a time, and they're also here rotating to sort of fill the space that hasn't been filled yet. From a leaf perspective, from a plant photosynthesis perspective, that's a good strategy for getting light in places where you weren't already getting light. Or another way to look at that is that the new leaves don't overlap the previous leaves. So it's effectively rotating the plant, rotating the leaf surface area so that it's collecting new pieces of light. And then over on the right here, this is actually a pretty common pattern in plants. This is what we call spiral leaf arrangement. And this is one leaf at a time, but the pattern where those leaves initiate is ends up being at a rather predictable angle from previous leaves. So I have this fancy term up here, phyllotaxis. This is Greek origin. So phyllo for leaf and taxis kind of for arrangement. talks about how the leaves are arranged along the stem. So this is one at a time, or alternate. These are two at a time, or opposite, and these are three at a time, what we call whorled. And then spiral. This spiral is a variation on alternate. So patterning, very useful for identifying your plants. And then in this class, we focus a little bit more on, well, what is it? What process was it that gave us that pattern? So some different pieces of evidence that tell us why we have that pattern. We're going to focus on this spiral phyllotaxis pattern for a little while. So what we typically find is that if you go from one leaf to the one before it, there's this angle of 137.5 degrees, which is the golden angle. This is something that philosophers and artists have been pursuing for a while, making spirals, finding spirals in nature. This is a rather common set of angles you find in plants. So we're going counterclockwise. I don't know if it's more likely clockwise or counterclockwise. But we'll kind of look at the pattern here, and the pattern ends up being that any one of these leaves is being produced at a location that's far from the previous leaves. So if we look at, say, 2 relative to 3, 2 is pretty far away from 3. It's not exactly across from 3, because it's also being influenced by the next older leaf, number 4. So right across from 3 would be here, and it's being influenced a little bit by 4 and kind of pushing it into that angle where it's a little bit offset by the 4. So what causes that? Well, sort of a combination of hormone. So this is a distribution of the auxin hormone that helps plants to grow. It's a combination of that and also sugar. So just kind of the nutrient availability. If these leaves are being made, they're kind of energy intensive to make these leaves. And so leaf number 2 here is being made at a place where the sugar, the resources haven't been used up. And it ends up making a pattern that is also really good for catching sunlight. So over here on the right there, that's going to keep playing for a while. What that shows is a simulation of if you were to arrange leaves in a variety of different ways. It shows how they work kind of one leaf with the rest of the plant. And some of these arrangements, the ones that the video stops on, some of those have the leaves overlapping each other like perfectly. So the ones where the light capture is rather low, let's see, we get one here. There we go. So that's pretty low light capture because that was 4 leaves that are all covering the existing leaves. Here's 3 leaves all covering existing leaves. Those are points of low light capture. This one, that one is a case where there's a whole lot of leaves taking advantage of light at different places where they're not covering existing leaves. So that place where it stopped, sorry, I don't have this at a option for stopping. So I'll just have to call it out when it happens. So it's going to get to the golden angle of 135 degrees, 137.5. Stop here. All those leaves, bunch of leaves made, and they're barely covering each other. So kind of an interesting interplay between the necessities of the cells that are making leaves. The leaves are made at places where there's resources available for them. That ends up being also a really good strategy for capturing light if you've got parts of the plant that are overlapping other parts of the plant. So this central image here on the top highlights it pretty nicely. We can see the golden angle, the spiral features of these leaves. You can see by the size of the leaves which ones are older and younger. And they're filling in to kind of fill in all of the local space that any area that wasn't already capturing light, the newer leaves are going to be able to capture the light there. So this is one strategy. This is a very common strategy. Lots of different plants have converged on this golden angle arrangement of leaves. But there are other strategies as well, including this one where the stem kind of does a little spiral thing. This one has leaves one at a time, but they're directly across from each other. This is a good strategy if you have, say, a like a horizontal branch on a woody plant, a shrub or a tree. Also a pattern we see pretty commonly. So I mentioned that the distribution of the new leaves is down to hormones and nutrient availability. There also are genes at play here, proteins that are involved in kind of signaling to some cells that they're going to become a leaf, and signaling to other cells that they should just hang out and continue being part of that dome of the meristem. So just sort of a, we can look at that pattern of leaf initiation a few different ways, and we get the same pattern. So it's sort of coordinated across a few different levels. So genes and proteins, enzymes developing based on nutrient availability, based on light availability. Sometimes there are leaves that are compound, and just while we're talking about leaves being initiated, it turns out that simple leaves and compound leaves have pretty much the same origin. So I'll direct your attention down here to tomato versus eggplant. These at this growth stage look almost identical. So there's a little bit of meristem here, there's kind of that dome there. And then this leaf on the left is the most obvious leaf. And then as it develops in the tomato, that leaf branches out and becomes compound. So this whole thing is one leaf here. In the eggplant, that just stays together as one leaf without any division. So this would be a simple leaf in the eggplant and a compound leaf in the tomato. These are related plants, if you were wondering. Let me